Hello, everybody. In the previous lectures, we discussed the major cell types involved in innate immunity, as well as the physical and chemical barriers of innate immunity. However, the cells that we talked about face an important challenge. Before they can respond to an infection, they must first recognize that as a threat. In this lecture, we'll examine how innate immune cells detect pathogens and tissue damage, how they communicate with one another, and how these signals lead to inflammation and pathogen elimination. By the end of this module, you should be able to describe how the innate immune system recognizes pathogens and tissue damage, and explain how immune signaling molecules coordinate inflammation and pathogen elimination. All right. So the innate immune system relies on receptors called pattern recognition receptors, or PRRs. PRRs recognize two major categories of signal. The first category is called pathogen associated molecular patterns, or PAMPs. These are molecules commonly found in microbes, but not normally found in host tissues. Examples include lipopolysaccharide, or LPS, from gram-negative bacteria, peptidoglycan from bacterial cell walls, flagellin from bacterial flagella, and various forms of microbial nucleic acids such as viral RNAs. The second category is called damage-associated molecular patterns, or DAMPs. These molecules are released when host cells are injured or destroyed. Examples include ATP, mitochondrial DNA, and uric acid. By recognizing PAMPs and DAMPs, innate immune cells can detect both infection and tissue damage. Once immune cells detect a threat, they begin communicating through signaling molecules called cytokines. Cytokines function as the language of the immune system. Some cytokines activate nearby cells, others stimulate cell division and differentiation. Many regulate inflammation by increasing or decreasing immune activity. Some help coordinate tissue repair after damage has occurred. Without cytokines, immune cells would act independently and immune responses would be much less effective. Instead, cytokines allow millions of immune cells to work together as a coordinated system. Although there are many cytokines, four groups are especially important. Interleukins are signaling molecules that allow white blood cells to communicate with one another. They help regulate many aspects of immune function, while interferons are particularly important in antiviral defense. Cells infected by viruses release interferons that warn neighboring cells and help establish an antiviral state. Chemokines function like chemical beacons. They create concentration gradients that guide immune cells to sites of infection or injury. Tumor necrosis factor, often abbreviated TNF, is a potent inflammatory cytokine. It helps initiate inflammation and recruit immune cells to affected tissues. Together, these signaling molecules coordinate the immune response. Inflammation is one of the most important outcomes of innate immune signaling. The process begins with PRRs recognizing PAMPs or DAMPs. This recognition triggers cytokine release. Those cytokines then promote vasodilation, which increases blood flow to the affected area. The increased blood flow contributes to the redness and warmth associated with inflammation. And cytokines also increase vascular permeability, allowing plasma proteins and immune cells to leave the bloodstream and enter into the tissues. This contributes to swelling. Chemokines then recruit neutrophils and monocytes to the site of infection. Finally, these cells eliminate pathogens through phagocytosis and other antimicrobial mechanisms. Although inflammation is often uncomfortable, it's a critical component of host defenses. The complement system consists of a group of proteins found in the bloodstream that work together to enhance immune defense. Once activated, complement performs several important functions. First, complement proteins can coat pathogens in a process called opsonization. Opsonized microbes are easier for phagocytes to recognize and engulf. Second, complement promotes inflammation by generating signaling molecules that recruit and activate immune cells. Third, complement can directly kill some microbes through the formation of membrane attack complexes, or MACs. The MAC creates pores and microbial membranes, leading to cell death. Finally, complement helps clear immune complexes and cellular debris from the body. Complement is therefore an important amplifier of the innate immune response. All right. So, in summary, innate immune cells recognize pathogens and tissue damage through pattern recognition receptors that detect PAMPs and DAMPs. Once activated, these cells release cytokines that coordinate immune responses and recruit additional immune cells. These signals trigger inflammation, which helps concentrate immune defenses at sites of infection. Complement proteins further enhance immunity by promoting opsonization, stimulating inflammation, and directly killing some pathogens. The key idea is that innate immunity is not simply a collection of individual cells, it's a highly coordinated system that depends on communication and cooperation among many different components. So, with that, thank you for attending this lecture, and we'll see you in the next one.